Biomarker development emerges from understanding the biology of cancer. So what it is on a practical level is really translating what we understand about cancer into the clinic.
We really focus on specific oncology-related biomarkers, and we think of them in several categories. So there are what we call pharmacodynamic biomarkers, and those are basically biomarkers that are used to show that you're actually hitting the target you think that you're hitting. We also are quite interested in predictive biomarkers, and these are really the raw material for companion diagnostics. So this is some characteristic of the tumor that really is going to allow you to predict which patients will respond.
We have three major subgroups within the department. One is focused on early biomarker development, what happens to a drug when it first goes into the clinic. The late-stage group focuses on biomarker development in all of our late-stage trials, in our medical affairs trials. And our third group is focused on companion diagnostics development.
There are certain areas that we focus on at different stages of development of a drug. In these phase 1 studies, what we really look for are pharmacodynamic biomarkers. These are biomarkers that change with drug treatment, and they can also have a dose-response relationship. In the setting of a phase 2 trial, what we really try to focus on are predictive biomarkers. Once we've identified a biomarker that's predictive of response to therapy, we then take that biomarker and test it prospectively in larger phase 3 trials. And that's generally done with a companion diagnostic.
This is really a phase where we're working a lot with doctors out there to really think about what does this mean in a clinical practice situation. So what we have realized in the last 10 or 20 years as researchers and scientists is that many diseases are very heterogeneous. That means if you look at 2 or 3 patients that we're saying have lung cancer and we do actually research on their tumors, we actually see that these individual tumors are different. And that also means that these tumors have to be treated different.
For a particular type of cancer, you can actually subdivide that into very discrete and separate subsets. If you have that understanding, you can then perhaps better target them with therapeutics.
We're very interested in understanding what are the factors that are associated with resistance to therapy, as that allows us to now develop next-generation molecules that can be brought into a clinical setting to target these patient populations here that are traditionally refractory to current targeted therapies.
We've come to learn that tumors have an evolutionary mechanism to escape points of inhibition. And through biomarker research, we're able to really understand what those adaptive mechanisms are and think about combinations of agents that address not just the initial driver mutation, but the tumor further and further out in time in terms of its clinical manifestation.
The impact of our work in the field of oncology in general would be that a patient would walk into a clinic and either provide a blood sample or tissue and get tested for biomarkers or diagnostics that we know are actionable.
Can you get a real-time look at circulating tumor cells or circulating tumor DNA and do that longitudinally throughout the course of treatment and really try to learn what are some of the changes that are going on? Is there an obvious resistance mechanism that would allow you then to potentially switch therapies now that the old therapy is no longer working? You've seen this new change, now you can give them a new therapy.
I think one of the exciting things from a companion diagnostic perspective is developing the requisite technologies that will enable those kinds of treatment decisions by integrating many disparate tests into one single platform that physicians and patients can use to make decisions.
We're very committed to making sure that the right patient gets the right drug. We know that that's going to take a different diagnostic paradigm. We're going to have comprehensive diagnostics, assessment of whether a drug works, continuous monitoring. What's really exciting about being here is we're supported to do that, and we have a role to play in developing that future.
If we want to make better drugs, we want to do meaningful work, and we want to have profound impact on the lives of patients, and we do think that our science and what we do here is able to do that.